Okay, let's hope for the best. Okay, thank you very much. So thank you, Edouard. Thank you, Bertrand, for the introduction. Yes, so I have been working in the Mechanobiology Institute for the last 15 years, and now I'm moving back to Marseille, well, starting on the 1st of Jan. So I'm super happy to meet the community here and start discussing with you of what we can do together. So I actually changed a little bit the focus of the talk and make it broader than what I expected at the beginning. And it won't be so much of a brain strain. I will reduce as much as possible the biology. And what I wanted to tell you about is how in the last 15 years, we coupled microfabrication to imaging in order to address different problems in mechanobiology. So it will be more of a catalog of different devices that we built and show you how they interconnect with one another and where we can learn from them. So the first thing we started with 15 years ago was try to image cell-cell contact and understand how forces shape cell-cell contact. And the first thing we did was relatively simple. We create microfabricated device in the form of microwells. And the idea was to stack two cells on top of each other in those microwells so that you can do an in-phase imaging of the cell-cell junction. So in this case, of course, when you do confocal imaging of that junctions, you get a much better, okay, yes, a much better Z definition of the system, and this is what you see here. You have the bright field, and now you have the two cells on top of each other, the green and the red, and you see how the junction forms, and the caderines are locating at the edge of the junction. That was really the best imaging at that time of cell contact, and now you can correct for all drifts and look at the expansion of the junction in in the plane of the junction itself. And with this, we could measure pretty much the dynamics of opening of the junction and so on and so forth. But not only that, it was also that we could follow in time how the level of caderine were varying with time. And you see here, this is the junction, and it's not the cell rotating, it's literally the local location of recruitment of caderine. And with this, we could correlate it to the local contraction of the junction. So the junction is not stable, it contracts on one side and not on the other. And as the junction contracts, the caderine are recruited locally. And we correlated this to the fact that the contraction of the junction leads to a slowdown of the actin dynamics, and the actin dynamics then leads to the recruitment of the caderines. So that's a spontaneous effect where the cell spontaneously have this anisotropy in contractility. But in the second round, what we wanted to do was actually to shear this junction or compress it. So the way we did it, we created this system, where you have in top chamber, you have this cup-shaped hole here with a tiny aperture at the bottom, so that the top of the hole, that's a tiny aperture there, and underneath you have a flow chamber. And when you have that, what you can do is you can aspirate a single doublet, cell doublet, from the top, squeeze it in until it reaches the center part. So you have one cell on the top, one cell at the bottom of this opening. And now once you have that, you can apply underneath, with a lateral pressure difference, a shear stress on the cell. So you're shearing your cell left and right, left and right. You can do this many times. As you see here, you have this configuration, top cell, bottom cell, and then you shear. And when we do this, you can couple it, because it's very close to the image, to the glass slide. You can couple it to high resolution microscopy, and you can look at, as you apply an external stress, how much proteins are recruited. Here, caderines, but we looked at different components of the cell-cell contact. And we could show that in this case, it was really the shear that would enhance the recruitment of the caderines, whereas the compression was actually not, was repressing the recruitment. And that was linked also to a change in the turnover of the actin structure. So that's external mechanical application that you can put on the cell contact. So then, once you have this idea of, oh, by the way, I forgot to mention, I put the main contributors of all the work here. I'm not citing them, but I think that's super important. The other question we ask is, okay, now the junctions are formed and they have this mechanical recruitment. Can we understand how strong the junction is? And in the field, the main way to look at how strong the junction is to take two cells at best and pull on them and measure the maximum force that the cell has to actually can undergo before they break. However, in soft matter, that's not how you measure adhesion strength. The adhesion strength or toughness of a cell-cell of any contact is actually measured by how much energy do you have to spend to detach cells, and has hardly anything to do with how much maximum force it can sustain. Maximum force is just one parameter of it. So in order to determine this, what Sri has devised is this comb of cantilevers, which are horizontal. And by virtue of microfabrication, we can change, of course, the bending rigidity of all these cantilevers. And at the tip of each of them, we can place a cell doublet. So now you have about 50 cell doublets in parallel. And you can come with a pipette and pull on one cell and measure at the same time the strain, which is how much the doublet elongates, as much as the force that is exerted on the cell doublet as you pull at a certain rate. So here you have access to what is called the strain-stress curve, where you have stress and strain that is needed to break the contact. And surprisingly enough, what we found was that depending on how you manipulate the cortex of the cell, depending on how you manipulate the amount of branched actin versus linear actin in the cortex, you can have very, very different behavior in the way the junction breaks. Here, you can have some cases where the cells are extremely ductile, and this is when you have a lot of branched actin, and now you pull and you see how much the cell can elongate. And in other cases, when you make the cortex extremely brittle, in the sense that it is very elastic, if you wish, but now the contact does something. Okay. So you see the contact breaks, snaps, and the cell's hardly defined. So we were able to actually create a phase diagram. We're looking at how ductile, having a proxy of how ductile the cortex is, or how tense the cortex is, and we can measure in this context all the amount of energy, maximum force, and maximum strain that you need to break a contact. Meaning that just looking at how much force you need is a lot irrelevant in many cases here. I'd be happy to discuss all this with you if you're interested. I don't want to go too much into the details. I like this one very much because you can also couple it to confocal imaging and look at this cell. This cell is my favorite. You know, there is one cell here, another there, and you see how much you can pull on that one before it breaks and making these tiny fibers. So you can completely change the mode of cell deformation and the mode of cell breakage. And of course, we have now the equivalent of what is going on here in cell monolayer, and we can check how that affects cell motion in MDCK monolayers. Okay. Now, this is how much force you can exert on the contact and how much energy you have to spend on the contact before it breaks. The other part, as Bertrand said, is how do you measure forces that cells exert on the substrates. For those who are a little bit in the mechanobiology field, I'm sure you heard about traction force microscopy. That's sort of the tarte à la crème thing. But one thing we wanted to do was to say, if I have this textured surface, which are made of pillars, that you want to measure the deflection of which will tell you how much force the cell locally exerts, can you actually image through these pillars to see the molecular structures that are on top of them? Because optically, it's a nightmare. You have these PDMS pillars, very dense one 1 micron or 2 micron in diameter, 5 to 10 micron in height. So imaging through this is very, very scattering. So at best, what you can do is usually measure the top surface and see how the top surface deflects. But as soon as you try to enter into the cell, you see nothing. So one way we tackled the problem was actually to change and work with a company in Israel that is called MyPolymer. And with them, it's not BIP, it's bio. Okay, I don't know why they all becomes. So we developed this polymer, which is with an index match, which is index match with a cell culture medium. So it's an elastomer, where it has the same property of the PDMS, except that its optical index is 1.34, which is exactly what is the optical index of the cell and of the surrounding medium. So here you have the comparison of how you can image cells at different magnification, either on glass, on PDMS pillars, or on MyPolymer. And you see that you can have absolute loss of contrast, even if you had a teeny bit of sucrose in your solution in order to match completely the pillar index. And in this case, pretty much you have your cells on pillars and the pillars are invisible. So if they are invisible, in principle, you can do super resolution imaging, and this is what we did with Sasha Peshatsky here. What we did was we imaged the flow of actin and myosin. Let me guide you through if I can click on something. Yes, okay. Let's see. Okay, so this is a close-up. You have in green here, you have the alpha-actinin. In red, you have the myosin. And you see how you have this retrograde flow. It's a bit slow. Okay, no, you see nothing. Okay, so you see how you have this retrograde flow of alpha-actinin and myosin. And this is done in super resolution with a structured illumination on top of the pillars. And as you see this, you can actually measure the deflection. Oh, we'll manage, huh? Okay. Yes. So you can actually, super bumpy, but you can actually see the deflection of the pillars underneath those cells. So pretty much index, yeah. Index matching the polymer allows you to image really through it without any distortion and allowing super resolution microscopy. So once we have that, although it's not very in the chronological order, once we have that, we decided why not coupling the idea of the microwells with the idea of texture substrates to make a combinatorial way of creating macroniches around cells in order to look at how cells will differentially react when they are in different microenvironments with different physical properties. So the way we did it, we call it memwell. We create a membrane full of holes that will constitute pretty much the wells into which we will put the cells in. So we can range any size between single cell to organoid size. And this membrane, you can separately coat it with different proteins on the side. We have a process by which you can even have the structured coating of proteins on the side, typically extracellular matrix, fibronectin, hydrogel. And once you have this membrane, you can actually glue it on another substrate where now you have also texture substrate or other kinds of coating, which allows you to create a 3D, quasi-3D environment where the side, bottom, and even top, if you add a layer on top, are different proteins or different structures. So we use it for many things. One of which was a crazy idea to say in epithelial cells, cells would polarize, with the apical basal polarity. Can we actually fool the cells so that they polarize with the apical basal polarity, just being one cell into the wells? Okay, so one cell only into one well, it fills the edges, and then let's see if we can find a sweet spot where the cell would polarize apical basal polarity, apical basal polar, whatever. That works, and it works best with primary cells, and especially with hepatocytes, which are the cells from liver. And when we placed single hepatocytes inside these chambers with extracellular matrix on the bottom and cadherin on the side, we could see this very strange structure here it's actin in green, and then you have this strange structure that appeared on the side of the wells where the cadherins are. And we wondered if that was not the equivalent of the apical pole that the hepatocytes would create, that are called the bile canaliculi, pretty much the apical pole where you secrete bile. Of course, now you have, even though these systems are in my polymer, that's why you can do a nice imaging of what is the interface between the cells and the substrate. It was not very practical for imaging either because it's in the vertical direction. So again, we decided to flip it and change it by 90 degrees. And in the end, what we did was we actually made these cells attached to a cadherin substrate and have extracellular matrix on the side. And now what happened was that, as you see with time, you see this actin structure. This is a single cell. Actin structure first at the edge of the cell that eventually goes into the middle of the cell. And when you look at it with all the proteins and even in RICM, you see that this region here is the apical pole into which the cells start secreting, and the RICM here with the interference patterns show you that this apical hemilumen, hemisphere, pumps up and down according to how much bile is secreted. So you can fool these cells as being in the liver and start secreting just by playing on the geometry and the display of the different proteins around them. Surprisingly, we wanted to know why in the liver, these lumens, they actually form into tubes. But in this case, they were always round, right? We had round cells and the lumens were round, sounded reasonable. We asked, okay, what if I change the shape of the cell now? Because these cells can be elongated and so on. So we changed them by patterning these caterines. So we had this oval shape, rectangular or square shape, triangular shape. No matter how much we changed the shape, we had always the same round lumen, showing that, okay, there's something that the shape of the cell does not dictate the shape of the lumen underneath. But maybe because it's in 2D. So what if we move to 3D? So when we moved to 3D, what we did was we took these macro wells and we used different geometries where we have the side of the macro wells coated with fibronectin as well as the bottom, or we have the side non-coated and anti-fouling and only the bottom that is adhesive to the cells, or the side that is non-adhesive, so the bottom that is non-adhesive in green and the side that are adhesive. And we stack the two cells in it, so you see it's coming back from the old idea of stacking cells. And depending on how the cells would adhere to the sides of the wells, we ended up with these structures between the two cells, which are bona fide bile canaliculi, so these lumens where the bile is excreted. But we can play with the shape of these canaliculi depending on the shape in 3D of the cell sac, as well as the way the cell adhere to the walls. This one is the one I like most. Here you put the cells in these triangular wells. You have two cells on top, and you see that the bile canaliculi, the lumen in between, extend into the three-lobe shape, pointing each of it towards a vertex. So we developed a little mechanical model to explain this. But just to show that this is linked to tension, the moment you release the tension by adding blebistatin, now all your lumens become round. There is no tension anywhere, so you might minimize the energy by having round lumens. So with playing with this system, we were able really to check how the forces and the spatial distribution of forces shape lumens. And this is probably why it was not working. Okay, never mind. I don't know what this is missed, but oh yes, I know. Okay, you missed the best part. Sorry, this is your part actually, Remy. Okay, let's see what I have here. Okay. So, okay, I'll just do that without the introduction before. So the other idea was, now, what if you have these micro wells, and of course you can couple it to these different imaging modes that I showed you about, but what can you actually do light sheet microscopy inside these micro wells? Ideally to do single cell localization microscopy or just light sheet microscopy. And of course, the micro well is not the preferred structure if you want to do light sheet microscopy. So about 10 years ago, we thought that maybe the side of the microwell can be made as mirrors. And if they are made as mirrors, you can bounce the laser on the side of the microwells and then have the laser illuminating a light sheet orthogonal to that. So I'm sure you've probably heard about SOSPIM already. So this was developed with Rémi Galland here and Jean-Baptiste Sivarita. And now you can do single cell imaging of the cells single molecule imaging and do single molecule imaging in 3D to do high resolution reconstruction of cell volume. Now, moving forward, we decided that maybe single cell and single molecule, I had a hard time convincing Jean-Baptiste that single molecule is not everything in life. So I told him, okay, maybe we can move to larger systems. And what we did was we extended the shape and the size of these tiny cavities to full organoid cavities. So now what we have here are thousands of tiny cavities which are pyramidal in shape. And what we do yes, the next slide is not working, so I'll tell you just how it works. You seed the cells in from the tiny opening, single cells or chunk of cells or chunk of organoids. You let them aggregate in, they form organoids. And now because you have these tiny mirrors on the side, you can actually do high content imaging of the organoid because you have each organoid sitting next to 4 mirrors for which you can use to do 3D imaging in a very fast and parallelized manner. So that's the best part, but it seems to be kept secret by Windows. And now you have, this is one of the of the thing we have. So you can change the shape of the wells, and you can image organoid at the resolution of 300 nanometer pretty much. These are small organoids, these are larger organoids, and you see that you can image even mitosis within the organoids, typically that are around 200 to 300 micron in diameter. And of course, there's no miracle here. You image pretty much up to 150 micron in. It's not that I want to keep the way it works secret. I will show you, if you want later, the functioning principle, but it doesn't seem to show up on the screen here. So in the end, what we do is we have these very large tape. It has its life on its own. Okay, good. We can have this virtual plate where we collect 96 organoid in about 1 hour in 3D. So it's 96 organoid, 3 colors, 70 plane in 3D in 1 hour. And with this, we can do AI analysis of where with Florent Levet, for example, or where the nuclei are and the shape and different kinds of structures that I'd be happy to tell you about. But pretty much that's the idea of how you play around this idea of microwells and make them, compatible even with optics and compatible with light sheet microscopy and high content screening. Now, a last thing that is missing in the field of organoids is organoids are great if you let them develop, but the problem is you want, ideally, to standardize them. You want to standardize their growth. And so pretty much you have to scaffold them a way or another. So usually people, they add extracellular matrix to it, but that's a very coarse way of scaffolding them. So one thing that we were thinking is, can we also tap on colloidal, because I come from the colloidal field, can we tap on the potential of colloids in order to play and create scaffolds? So one thing that I'm going to show you here is how we created this scaffold for bile duct organoids. Bile duct, when they occur in your liver during embryonic development, they start developing along a vein, which is a cylinder, right, with some shear stress in it. And then you have the precursor, the hepatoblast, so the stem cell that surround the vein, that locally differentiate into cholangiocytes, and these cholangiocytes starts forming lumen, a bit scattered everywhere, and eventually these lumens collapse or anastomosis and form this long bile duct. Okay, but when you try to recapitulate this system or this process in vivo by using either hepatoblast or cholangiocyte, this is what you get. You get beautiful cysts, beautiful spheres, but far away from anything that looks like a tube. Okay. So the question was relatively simple. Can I actually change the vein here by just a gelatin tube onto which I will graft the growth factors that are able to drive cells from hepatoblast to cholangiocytes? The idea of that is, if you can do this is what we did actually, okay? But if you can do this, you can make thousands and thousands of these rods and have hence the capability to do scaffolds in high content volume. And that's what we did, and I want to be fast here, but you see that along the rods, you have these, that's at day 1 after seeding, you have these cells in green, which are the cholangiocytes, and they only differentiate in the close vicinity of the rod, and they're surrounded still by the hepatoblast. And as time goes on, you see that you have the development of tiny lumens along the rods here, and eventually they merge into long tubes. Okay, now these things in terms of size is a nightmare for optic because it's 500 micron in diameter and 1.5 millimeter in length. So it takes about 10 hours with a 10x objective to do this sort of confocal imaging, notwithstanding the fact that you have to do 3D reconstruction, and I lost a grad student on that because she quit. But hopefully I managed to get her back in the project by, that was the most important one again, okay. No, so you missed this one. Okay, never mind. I'll tell you then, sorry. What we did was we liaised with a group that does optical coherence tomography and in multi-well plates. And when you do this, you have in 20 seconds, you can image completely your whole organoid along the vein with the idea that the only thing you're interested in are how the tubes elongate, what are the tubes, and you don't care about the cell. The cell, you leave it for analysis with confocal microscopy, but now you can look at the dynamics in 3D of the growth of the tube. And indeed, you have very high content imaging of the of the lumens after OCT, 10 seconds per organoid to image in 3D, and then you can train an AI model to segment only the tube. So that's the one plane of the OCT. This is the rod in the center. The white here are the cells, and the green are the segmentation of the lumens that now you can reconstruct in 3D. And when you reconstruct in 3D, the codec is indisponible. Okay. So when you reconstruct in 3D, you have these lumens here, and then you can see how they connect together, and you can follow the dynamics of their growth. And this is compatible with a high content screening. All right, so I reached the end. I hope the thanks in the end are not half, okay? They're missing some parts. So this is 15 years of my work at MBI. There's a lot of student postdocs, and I haven't cited everyone here to thank. But I really want to thank these main collaborators, Gianluca, who is the head of the microfab facility at MBI, with whom we worked very closely for these years to develop all these systems together. Rémi Galland here, and Jean-Baptiste, with whom we have collaborated for 10 years also on the development of SOSPIM. And I'd like to thank you for your attention.